Canine Detection Collaborative. We bring the conversation straight to you. Training tips and helpful hints you'll actually use. Real insights from real experts with a side of laughs. Canine Detection Collaborative is where it's at. It all comes down to just one thing, helping our dogs be the best they can be. Canine Detection Collaborative.
Hi, everybody. Welcome back for part 2 of our interview with Dr. Jenny Essler. Stacy and I and Dr. Essler pick up with some of her really interesting research, where she wants to go, and the super exciting announcement at the end of the episode. So make sure you listen all the way to the end. I hope you enjoy the episode.
Canine Detection Collaborative.
So what type of research are you doing now?
So a lot of my research did pause a little bit when I moved here because I had to prep all these classes. Anyone who's in academia or you guys that run classes, it's like full and a half time job prepping a class, which I had never done before. And interestingly, no part of a PhD preps you at all for making syllabi or classes or anything.
No, they just want you to teach you how to do research.
Yeah, like I do research and now someone's like, now you have to teach these students. And I'm like, that's not really the same thing, but all right, I'll figure it out. So I did that had to take a little bit of a back seat. And then most recently, the work that we have been doing, what is really trending towards invasive or imperiled species work. That is largely because on campus, on top of having this really unique canine program, we also have a really unique fisheries and wildlife program. We have like a full hatchery on campus. We have students.
I heard about that, by the way, from
it's amazing.
Yeah, they searched it, Robin.
Yeah, you searched it?
I think so. I think they were talking about the fish tanks. Weren't they talking about the fish?
Yep. And so this collaboration came with a student that I had a couple of years ago, Katie Baranowski. She's a PhD student now at Albany, where we were like, okay, what's an invasive species? That's kind of I don't want to say hip, but like the one everyone cares about, right? Like that's how you get money. Everyone cares about a species. And they said the Round Goby. So it's a fish. So we had the Fisheries and Wildlife Program do the fish side, the collecting / housing. And we did the dog side, the training / teaching of students. And the other difference here compared to Penn, it's all of my research really has to include kind of like teaching at the same time. So, at Penn, I had students, but here I have like students, like a lot of students, and not necessarily students that want to go into science. And so they're really focusing more on like the handling, learning for application side, more so than learning to like get a grant and go teach it side. So 2 summers ago, we started this Round Goby project with, I think, 6 dogs. One had to drop out. And it was literally just like, can you find the odor? And this is one of those things where everyone asks, like, have you ever tested a dog and they couldn't find the odor? No, of course not. I've never found an odor they can't find. But that's not how academia works, right? You have to prove it.
Right.
So that was the first one that we did. And we compared them to 2 other fish that they had in the labs, 2 other fish that are found in the same area. So it's not just, can you find a fish, but can you find this fish? And it was just presenting the fish to them. So we weren't at the point of just using like water, which is kind of like what the eventual goal would have to be. And they did great. Some did better than others. Interestingly, and this is another like side road we could take in discussion. One of the dogs, we have one dog that we purchased from the Penn Vet Working Dog Center, who is an amazing detection dog, but had some like minor back issues that would cause him to, he takes gabapentin, basically, he's fine, but no one's going to buy a dog that needs meds, basically. He did not do as well as some of the others. And I think it's because despite his pretty extensive and amazing search experience, he did not have a lot of experience discriminating between highly related odors. So he had worked on UDC, he had worked on live human, he had done smokeless powder, but like my 2 that were in it had done COVID versus non-COVID, and they had done.
Oh, right. Very laboratory level discrimination type of stuff. Yeah.
Much more like smaller differences. So even though he is like for sure a like amazing and in some ways better detection dog in this task, that prior experience not being there really like affected it. So he got there. It just took him longer to get there. He had to pay more attention than he probably ever has. So it was less presence, absence of odor and more like nuance of like different but similar odors.
Very cool.
I went back to the whole we're presenting the fish thing.
Yeah.
Yeah.
How do you present the fish? Is the fish dead? Is the fish
Nope,
alive?
No.
It's a live
Like
fish.
in a tank, or were you like?
So the Fisheries and Wildlife Program kept them. We went and collected, I say we, Katie did it, not me. I shouldn't take her credit, but she would collect them for the day or the session, put them in little mason jars with like mesh top lids, so metal.
How big were the fish? Because I've got like in my head trout.
3 inches or so.
Oh, okay. So they're like
I'm picturing
beta.
like
No,
They're like betas.
yeah,
okay.
yeah.
I'm picturing like trout.
No.
Thank you. Thank you. I appreciate that. Because in my head, I'm like, you just hold the fish out and go, here, sit or
No.
Okay.
They're small.
Thank you.
So we just put them in, we made the tube, the wood with the PVC, the basic kind of setup. And again, I shouldn't say Katie made them. And
That's part of being a student, graduate student. You have to make all your own stuff.
When I was a student and someone was talking about what we did, I was like, that was me. What do you mean? Did that. I purchased the wood, Katie made it. And that was pretty much it. It was a very basic like odor recognition. Like, can they use it or can they do it? Can they discriminate from just water and then to other fish that are found in the same area in the rivers where they populate? And yeah, that's really where this started. And now we're at the point where they are starting to try. It's a little slower here because it, again, has to include like student learning. So it's not like just here's a handler with their amazing dog and you're going through body. Not that we don't have amazing students, but now we're at the point where they are starting to work more in kind of like real life conditions. So putting them in fields and outside searches type things. This semester was our first semester having a conservation detection dogs class. So the students that were doing detection, but like within the context of like working outside and working in different environments than like buildings, cars, but like really more of what you would see in conservation type setting. And then they were using the Round Goby as their target odor. So
my brain's
yeah.
Uh-oh.
like, oh no. No, so are they on a boat? Like, how are they? And then the thing is, like, the fish swim away. It's not like, like a body in a lake
that stays there.
Right. I'm picturing your target odors just swimming all over the lake.
Yeah. So there's kind of 2 end goals for this. My end goal as a researcher is, can I take water samples and present them to the dogs for presence absence?
Oh, okay. So presence versus absence.
Correct.
Okay.
There's no, at this point, it's kind of like the spotted lanternfly. There's no chance of eradication. It's tracking.
Oh, okay.
And so we're not looking to find the fish. We're just trying to track them as they spread. And that's what I'm working on. The other faculty members who are more actual handlers are working into, can you work them into that odor, right? So if I start somewhere on the river where I know they're not present, and then for that change of behavior, so they're looking for that application as much as to train students how to do that in the field, whether that's like the end application versus what I think the end application is. But yeah, it's never going to be like, here's a fish. It's just, are they here pretty much? Yeah. So it's very different than some other like conservation fieldwork settings where you're looking for scat or you're looking for feathers or something. It's not that. It's just for tracking. Yeah.
So what do the goby fish do?
They pretty much outcompete everyone else. They're very good at like modifying what they eat based on where they are. And some, just like there are some
carp, it's kind of like the invasive carp kind of.
Yeah. And that, part of it is just this, like a messing up of the ecosystem. They become quite prolific and they just take over what would be food for other fish. And they don't have as many predators as some others might. There are varying levels of, there are varying varying interpretations of just how bad it is to have them. Same like with spotted lanternflies, there's people that are like 100 out of 100 panic versus much less. So depending on what like ecologist you talk to, it might be a little bit different. But I think for us, it was a mixture of they're easy to get, they're local, we can train the students, and there is an end application for dogs. I will say since then, since it has become kind of, I don't want to say popular or public, but like everyone in the area knows we're doing this, we have been approached by some people from more government type organizations that work in conservation and mostly invasive species work to see if we can apply the dogs in other applications. So one is hydrilla. It's a plant. Crawfish, also invasive, certain type of crawfish. So I think the benefit of the project, even if we never get to the point of like, here's a goby fish, it has like kind of helped us make relationships for us and for our students to do other projects, right? And part of getting grants and all of this is just, have you done it before? And, I can do it here. And so give me funding to do other stuff. So yeah, we'll never be finding individual goby fish.
Okay. What about like eggs? Like, would you ever look for like the eggs or?
Again, I don't know if it's beneficial because we're not looking to eradicate them.
Well, I just meant like, and again, I don't know anything about like on boats or, that, I guess it wouldn't be on boats. I guess fish eggs wouldn't end up on boats.
The issue is that especially depending on the time of year, they go quite deep, and I'm sure we've all worked odors where it's very deep and that's just makes it much more difficult, especially with, it's fish and there's fish everywhere. But, the comparison for us is that the groups we were working with, so the Scott George is a researcher that we were working with who works for USGS. They use eDNA, right? Where they take a sample and they assess it for the DNA of the fish. It's expensive. You also have to wait for the sample to like go off, get assessed for the DNA and come back. And so I know that Nathan Hall and Paul Bunker and some others did a study on comparing dogs to eDNA, and eDNA is definitely more sensitive, at least for the species they looked at. The dogs are faster, right? Like I get eDNA right now. So I think it's as long as dogs have a benefit above like the current application or options, they're still a better option. Like if we had a perfect metal detector for bombs that didn't have any downside, dogs would lose their jobs, but
right. We don't.
Dogs aren't 100% perfect, but they're better than the alternatives. And so that's kind of what we're going for with the fish stuff too, where can they fill this niche where the current alternatives are just not as good or, more expensive or something like that. Yeah.
Do you know of anybody working dogs with beetles? I had somebody email me. Actually, we started having a conversation about American burying beetles, and they wanted it's a protected species, and I was just reading it, they end up overwintering and go nocturnal and spend days and overwinter underground. Mm-hmm. So I was like, what about this can you isolate about the beetles? So I was just asking because she's working with, she's getting her master's in entomology. And
so she is training dogs or just interested in anybody who does it?
Well, here's me. She's working, I'm trying to connect dots, right? Because she's working with researchers out of University of Oklahoma or Oklahoma State, Oklahoma State, who's the leading researcher on ABB right now to get his thoughts. And so I don't know where she's getting hers. Anyway, so literally their densities are really low, like one beetle per 500 acres.
Yeah. It's interesting that you bring that up because I'm not an entomologist or expert, but I believe I've had this discussion with someone on campus because they did have a grant to study this, not with dogs, just the Fisheries and Wildlife Program did. And I remember asking her, hey, wouldn't dogs be a great application? And she said, oh yeah, there's somewhere already doing that. And I would have to look through my emails to say whether she gave me that specific info or whether she could like put me in contact with that person. But the answer is, I think so, yes. But that's kind of the end of what I know. And I don't know any more than that. But yeah, I mean, it was the first thing I did when I got here. I looked up like, what are people studying on campus and how can I use dogs to also study what they're studying on campus.
That's fascinating. Yeah.
Yeah.
Yeah. Because we were talking about detecting their brood ball.
Yeah. When we first started the Goby project, that was our thought, right? We'll just take any Goby odor that works. I wouldn't be opposed to using eggs or just water from the tank. Like really, I think they should just be trained on everything. I mean, it's a level of generalization that I don't necessarily think is going to be an issue. You know, the issue would be if we're then trained on eggs, we now need to have control eggs, right? So they don't just think it's everything. So every different iteration of Goby we introduce, it does add a necessity for more controls, but I don't know that it's necessarily an issue. The ones we have haven't given us eggs, and I haven't asked them to go find eggs for me. And so I think that's probably just the only reason why we haven't done it. I can't think of like a specific reason why.
With the beetles, are they looking at it from like a concentration? Like if they're doing like I feel like the brood ball would be a concentration kind of high number.
Okay, so I'm reading from a Facebook message, right?
Because it's like, interesting.
Right. They had many challenges because the densities are low. Inactive beetles bury up to 10 inches daily. There are a number of other species that the dogs were unable to distinguish between.
Oh.
And so the broods progress rapidly 10 days from carcass to male, leaving third instar larvae. So that is above my entomology understanding.
Level of development.
Okay. And so their smells likely change. So literally, and then you have to have permits to dig them up and all of these other things. I mean, that's one of the things when I was working with a student from Australia, you know, she's trying to teach her dogs how to detect a certain type of orchid that grows underground and all sorts of other things is considered not highly dangerous.
Like, is it endangered?
Endangered. Thank you.
Endangered.
I'm like I and E.
Well, no, your brain went. I know where your brain went. Australia, dangerous. Because everything in Australia is dangerous.
Wants to kill you. Yes.
Right.
And so
I know where it went. I know where your brain went. I got it. I'm like, you mean endangered, and I know exactly why your brain went the way it went. So
we're talking about how do you collect odor for that when you can't collect the thing, because collecting the thing would kill the thing. And, you know, in this case with the brood balls, they evolve so quickly. How do you do that arrest in time feature? I mean, yeah. So it was just fascinating.
Think how those issues come up in everything, right? Like, how do you find each specific time point of HR that you need to train? Like, how do you catch it? No, there's always something, and it's all similar problem. We had the same thing this summer. All of our Goby died, not for any reason. They just die eventually. And you can't just go get them because they're invasive. And so you're not like allowed to go get them. And so we were at a point where we kind of had to pause until someone could go catch more Goby for us. So it's the same thing. You sometimes are really at the mercy of like whatever you're studying. When I did spotted lanternfly at Penn, we were getting eggs from the Department of Agriculture. They killed them for us. They heat treated them and then sent them to us because obviously they didn't want to send eggs out of the quarantine zone. But then by the end of the study, they were in Philly. And so I would just like walk outside and collect some eggs.
Oh my gosh, I'm here at the house. Like I open up the sliders. I'm like, get out of my house. Like you're kicking them outside.
So it was a problem at the beginning in terms of like, I don't have 100 samples for them to generalize on. And then at the end, it was like, how do I train outside? They're everywhere. Place hides. We even went behind the center. There's like it's not there anymore, I think, but it was like a kind of walkway. Either side had trees. And one of the dogs, Stacy, who was a USAR, human range dog. So she was amazing. Not the best lab dog because it's like bull in a china shop level of searching. Right?
We have dogs that represent that.
Oh my gosh. Yes.
So she was great. Sometimes her body / everything worked faster than her brain on like the scent wheel.
Sorry, I resemble that remark.
So we took her outside of this field and we're like, oh, they're everywhere. So she should be a like 10.1. But they were so everywhere that like she was in constant odor, right? And so saw this massive change in behavior, but then also frustration of like, I can't get closer. I can't get further away. And so that was our issue, right? That eventually was one issue to another, but a really nice learning opportunity for me, but also for all of us. It was the first invasive species project that we had done, first really biological study I think we had done, other than like the cancer stuff, which is like very lab, right? That's like pipette, plasma from a deep freezer on the scent wheel. So it was a really nice learning experience and has really helped with helping to implement this type of stuff for my students and kind of talk them through when we hit these walls, what are our options and, what also stuff too.
Total. Okay. Welcome to the not really rabbit hole, but you were talking about the cancer research.
Yeah.
Right. So what cancer research were you guys doing?
So when I got there, before I got there, they had trained a few dogs on ovarian cancer. So for those of you who don't know, ovarian cancer doesn't have any like test for it. So most of the times when it doesn't also show symptoms when it's early on. And so has pretty poor prognosis and is usually found later. So kind of a good opportunity for, can dogs help us learn something that can help make a test? So the goal was never, to my knowledge, from the time I worked there until now, was never to like have a dog in a hospital. It was a collaboration with our lab, Monell researcher, Dr. Preddy, who was there, a chemist, and then also a physics lab at Penn. Can all of these wings together make like an electronic nose that can assess blood? So can we train the dogs, test what VOCs, volatile organic compounds, are the dogs attuning to, turn that into a blood test? And then the other thing that it was always an issue for us is because it's a relatively, I would say it's a rare cancer, but we didn't have like a whole bunch of samples. Can we train them on cell lines? So just normal cell lines from a lab and have them transfer it to like plasma. So can you train them on basically, I don't know, the equivalent would be like buying a training aid and then hoping they generalize it to like all companies of smokeless powder kind of thing, which yes and no, mostly no. And it was the same for us. We had one dog that seemed to be able to generalize from the cell lines to the blood plasma, but it wasn't like a lot of them. And so we moved back to the plasma full time. And yeah, I mean, the end result of that was a little bit kind of, we just didn't get more funding to like keep going down that route, not to get political about it, but it is a disease that mostly affects women. And so there's less funding out there, but, I will say, and it's published, but we were able to present to the dogs, snips of that whole odor profile to see like which parts they were kind of seeming to pay more attention to. But as you can imagine, there wasn't like every dog chose this part of the odor profile to attend to. It was a little bit varied. But I do know that in the physics lab that was working on it, and I am so sorry that I'm forgetting the PI from that lab, I can't remember his name. They did do pretty good at using modeling and predicting samples. So I hope it's still going on in the background. It did kind of like fizzle out because of funding, unfortunately. It's a real bummer because it could have been quite good, and hopefully it is in the future. But, even if you think about like skin cancer, breast cancer, with all these studies, dogs can find it, but like, what do we use that for other than saying dogs find it, right? Most of these studies on dogs detecting cancer kind of end there. They don't take the next step of like, how can we apply this?
Total layman, don't understand anything question.
Yeah.
I guess the question that a lot of people might jump to is, why couldn't you have a lab with working dogs in it and have them actually do the test?
Yeah. Well, having done probably a bajillion sessions with these dogs, as you probably know, they're not on their best every day. And so there was that you would get false positives or false negatives. And that's a big tell people they do or don't have cancer. And even if they were perfect, right?
Yeah, no, that makes sense.
Dogs work, right? Not a year.
Like, what are you willing to bet on that outcome?
I mean, I dogs were amazing. I don't want my cancer diagnosis coming from any of the dogs that I worked with, because there were days that our superstar dogs came in and they were just off.
Yeah.
Right. There's no way to know that except for that I knew what the samples were.
Yeah.
Right.
Yeah.
So the goal of that project was always to get information from the dogs, date a more specific test.
Well, I think a lot of people will actually appreciate that answer, because I think that part is not actually that well understood.
Yeah, I think it is really important. We do put some level of kind of, I don't want to say like over trusting in dogs, but especially if you haven't worked with dogs like you guys have and some of us have, to actually see really dogs have really not good days. I think some people really just don't know that happens even with the very best dogs, right? And there's not necessarily a way. Sure, there's some days where the dog's handler is like, yeah, I can tell they're just off. And then there's some days where like, you can't tell, and then they're just off anyway.
They're being a dog. What are they doing? They're supposed to be this little robot on 4 feet.
Yeah, the little machine.
Yeah.
You know. Yeah.
Yeah. So I'm not getting diagnosed
Stomachache that day.
by dogs anytime soon.
They ate 18 eggs that day. Sorry,
Which she just called Dash out because Dash just
I called out Grandpa Dash.
Grandpa Dash just decided, well, Joe, the husband, was not paying attention, and Dash is over here while Laura Land's at work. And literally the bucket of eggs was sitting on the floor, and he just had a little old
fresh
from the chicken coop, shells and all. Literally, yeah. So he ate a whole bunch of them, and let's just say our house has been kind of stinky.
Yeah.
But my point is, if you asked him to then go out and do some
work,
I'd be like, not today.
But if you hadn't seen him eat those,
I wouldn't know.
Right. Prepared for him to have an off day, right?
Right.
And I know,
Sorry,
when
Dash.
you get deployed and stuff, it's always, you want to make sure that, your dog is doing their job, because you're you're in a service role for people, right? It's very serious. But also asking a dog to do it in a lab is kind of an equal level of serious that has less opportunity for us to say they're off, right? Because it's just searching a scent wheel. It's not on a rubble pile. It's not flying and driving. There's more opportunities in those scenarios potentially for you to notice a dog is off. Whereas it was grab a dog from the kennel, maybe you prep a couple minutes and then you're searching, like you're telling me if it's cancer or not.
And you don't know if they're not feeling great that day or if you didn't get enough sleep.
Yeah. And, the other thing is that it's very cool and it's also very boring for the dogs. Like
it is
so to search the scent wheel or search the olfactometer boxes. Like it's cool, but it's so boring.
Right.
And some of the dogs just stop doing well because it's boring. Like, most of the dogs we retired was not because they couldn't do it. It's because they didn't do it anymore. They're like, I am so tired of coming here and telling you where the cancer is 10 times in a row, twice a day for the last 5 years. Like, I told you where it is, lady. Like, I'm done with this task. It's not fun. And so that's, that's the other side of it, is you also have this living animal who has their own feelings and opinions on the thing you're asking them to do. And the nature of that task is that it's just boring, even though on a human side, it's amazing and impressive and super important. The dog doesn't know they're finding cancer and helping people. The dog is just walking around a scent wheel or walking around boxes, and there's
no hunt.
Yeah.
Well, and when we did the interview with Steve White, he talked a little bit about the difference between detection dogs and examination dogs.
Yes. That was a great way of delineating that.
Right. And so it's been very interesting. So to think about that. So now that you're getting to hopefully start up your own research, what's your research? What do you want to do?
For me, and we were talking about this very briefly, I know I'm really interested in the human side of things, the humans' interpretation of their dogs, the training, especially working with all these students. We put a lot of effort into how do we pick the best dogs.
Uh-huh.
But maybe we should also have some discussion on how we pick the best handlers.
That'd be amazing.
That I'm super interested in biases, even not in terms of calling people out, but how do we assess them? How are people approaching these things? I've been training these students, and just like probably you find in the real life version of these fields, people panic when it's like blind, right?
Yes.
I trust my dog, and then all of a sudden it's like, no, I don't actually trust my dog.
Well, and then you put certifications and all that stress on top of it. And, even I get weird.
Right. Yeah.
Yeah.
And so I'm really interested in that side. I am also really interested in, I would love to do more research on human remains type detection, because I do think, especially working with my students, like I said, these kind of like, what are students really interested in? They ask me questions. I kind of have to say, like, we don't really know. Like, we don't know all those answers on, how is it changing? How do you, what level do you have to train the dogs to get X level of discrimination or generalization? That's really interesting for me. I know we talked about, Robin, when you have that seminar, I'm like, how do I get to go there and research all of these amazing dogs that you're going to have there? Because you just never have that many in one place. And I would love to keep doing some of the predictive testing on younger dogs. When I started at Penn and worked with Pat Rolu, again, how do I turn what she's doing into something anyone can do when they're assessing dogs, right? How do I take that decades of knowledge and like write it down? We talked about this idea of, at Penn, very few dogs fail out, but they also have way more time to put into every individual dog. And so at what point is it like, I could train 3 dogs in the time it would take me to train this one dog? Like, where does that land? Where this could be a detection dog, but the amount of time I had to put into it probably isn't worth it. And what are the behaviors / trends that we see to pick that earlier? And it's probably field dependent, right? Like SAR, different from single purpose, things like that. But I am really interested in taking what a lot of practitioners are doing. And I don't want to say standardize it, but make sure we don't lose that knowledge, basically, for everyone.
It's so interesting because there's a whole bunch of stuff that is lost. When I was out working with Raven today, I was banging my head against the wall or my steering wheel when I got back to my car, because I realized I'd skipped a step with Belle that I had done with almost all the rest of the puppies.
Really?
Right. And it had to do with where I placed the hides in the stores.
Really?
Yep. And the hardware section with all those little tiny drawers are great for hiding bottle caps in, where they can get their nose on it, but they can't see it.
Ah, okay.
And I can put them right at nose level.
Yeah.
And then start varying them out to different heights. So they start learning to work it like the wall.
Yeah. But it's bottle cap.
But it's a bottle cap. And so it just became one of those, And talking to trainers and the people like Pat who do assess a lot of dogs. Well, okay, so they twitch a little bit here and you said no, flush them out of the system. Why did you say that?
Yeah.
What do you see that lead to that you said no?
Yeah. And the other hard part is that in these larger programs, what happens when you say no? They're gone.
Right.
So you lose any data from later in that dog's life that you can say, yeah, they wouldn't have worked out, or these are the things you see later. In a perfect world, you would keep those dogs and keep getting that data. And so you wouldn't have this ceiling effect of every dog I look at is amazing because we only keep amazing dogs.
Right.
But yeah, exactly. Those little twitches, those little small things, like kind of trying to qualify and quantify that so that it can be used by others and it can be used to streamline these processes better as you can, right? Again, they're dogs, not machines. So that to some extent, it is what it is. But having 2 dogs from Penn that didn't work out for them, but are working fine for me, like in a lab setting here, when could we have seen that? Like, when was the turning point of either of these dogs not being able to be sold and work for somebody? And you talk to the trainers, I talk to the trainers like, yeah, I saw that. This is not surprising to me. Okay, but why? and at what point, and what would have taken to get them there? Is it worth it? Or should you just walk?
To what degree is it also the trainer versus the dog? And I'm not saying it was in that situation, by the way.
It is general comment. With a different trainer, it would have been different. There's just something beautiful.
Yeah. And I'm kind of going back to like, we just had an episode, Robin and I recorded one, and we were talking about how when Prize had her litter of puppies, and I was freaking out over whether or not I thought Jack was I picking the right puppy? And, Robin's got tons of experience with this. And she's like, especially with what I want to do with him, she's like, he's going to be amazing. Relax. And it's like, what is it that, I was feeling like, yes, but because I was like having to sit down and like, yes, I'm picking this puppy.
Yep.
But then it's like, how do you then put a pen to the craft, right? And the experience around the craft part.
That when, as far as I know, almost every study on like personality and all of that in dogs is not predictable until way later, like 6 months plus. And so how is someone like Robin able to choose a 6, 7, 8-week-old puppy and be right about it?
But she is.
Right.
Yeah, I would like to say most of the time. It's not 100%. Most of the time.
Probably not 100%, but yeah.
But, how can these people do it? What are they looking at that we haven't yet been able to find on like the academia research side? People are doing this successfully, but we haven't replicated it yet. We can't predict it. And so.
And it's really interesting because I've developed my whelping practices, right, to be able to help me very easily determine some of that stuff.
And I could see a lot of that this spring.
Right. As we're watching, I'm like, okay, Stacy, this is what I'm seeing out of this puppy, and this puppy. And it was literally came down to, okay, guys, we need to watch this puppy and this puppy. I need to see which one is going to do this more.
Yep.
And then it became it doesn't matter because it's going to come out in the wash for some of the puppies, because, okay, I have 2 puppies who are going to go into XYZ homes and it doesn't matter.
Yeah. Yeah. So when I was at Penn, I also did the puppy testing / socialization for the litters while I was there. So I did 2, 3 lab litters. And I remember multiple times like having to give like feedback on like what I was seeing, what we were tests. And I think also the hard part is you're also, again, selecting for litters where like pretty much everyone's pretty good. You're looking like very small differences.
Yeah. It came down to, Stacy's going to giggle at me because I went, and when I picked out my little picked out Raven.
I'm already laughing because I know the story.
Right. They unfortunately, because this happens when you have a kennel, they had Giardia.
Yeah.
Right. And they didn't feel well.
Mm-mm.
And so I drive up there and I'm trying to pick out my puppy, who you can tell none of them feel well. And they're just like, okay, this is dumb.
Mm-hmm.
And my temperament test came down to the puppy that followed me with their tail in the air.
Well, my test was like for Prize. I had no idea what I was doing. I got very lucky, but it was a great litter.
Mm-hmm.
That's some of the things about genetics. Sometimes the genetics are just there.
Yeah.
I don't think I could have necessarily gone wrong. I think I did a great, I know I had a great pick, but it's like I saw something I liked and but the genetics were already there.
Yeah. I think and again, it's only 3 litters of experience. So it's not exactly like a whole lot, but
mine's 1. So,
the thing is, it's sometimes easy to say 1 or 2 are going to have issues, but then you have like most of them that are good. And like, for example, the last litter I was there for, which also was a COVID litter, sort of. So it was like, go over there, have a mask on. They were great. But one of them at 4 weeks old was terrified of heights. Like we put her on the table, like, and I had never seen that in like any of my, like the other 3 litters, just completely shaking.
Wow.
And so that was easy to say, to watch this dog, like, does this maintain? And it did to some extent. She was very highly motivated to work with people, never really that interested in like unstable surfaces, heights and stuff. And I'm lucky because I have been able to keep in contact with who's her handler now. And so a lot of that has maintained, at least like she had like 10 out of 10, like I want to engage with people, but like not probably going to go and rebel.
Right.
But most of the rest of them were like 8 or 9 out of 10 on both. Very few are like, oh, I'm not sure about this one. You can easily maybe cut a couple, but then you have like a whole bunch that are like, they have great genetics. They're all doing good. And it goes by history, the experience of the person picking, which I certainly was not experienced, but some of the whelpers we worked with. So before they moved, Tracy and Eric Darling were the ones that were whelping for Penn. And so Eric was the one I would talk to a lot about like what he's thinking about the puppies, because then I had to go back and sometimes we had to choose like which ones we're keeping, which ones we're giving. And I remember them asking me that and I was like, man, I haven't even worked with detection dogs or working dogs for a year. I can tell you what the puppy tests are saying, but so working with someone who had done that for so long really put of that nuance to it, that's really what I wish there was some way to put into words, that feeling that you guys get when you're looking at dogs. And you can say, this is going to be amazing, or, this is the dog I want. Well, if you want it, why do you want it?
Well, and that will be one of those things. Then at my next letter, let me know if you want. Usually this year we had Unleashing the Hounds.
Yes.
And so we would say, okay, every 15. And that's because Laurel Ann had come over and we wanted to watch the puppies because we would rearrange their playpen every night. And so every morning we would unleash the hounds, and it became part of the who attacked which obstacle.
Yeah.
Right. And who got scared and who took 10 minutes to do it and what was the most challenging thing that day for all of the puppies, and who just ran headlong into everything and didn't care. Jack.
That
was Jack.
That was my puppy.
Yeah.
Yep.
And that helps me stratify the puppies.
Yeah.
It got to the point where the puppies were like, this is too easy.
Yeah, this doesn't matter.
We could not make it.
You get that ceiling effect, right? Where you just have these amazing dogs and it gets harder and harder to give them for like gonna make them look different.
Yeah.
We can't make this harder without making it unsafe.
Dangerous.
And
right.
So we can't do that.
Right. So that's what we ended up doing. So, okay. I have a question for you, Dr. Essler.
Yeah. Hopefully I have an answer.
Well, you do. Because, in podcast land, we've already talked about this, but yeah. Would you be willing to come on a like quarterly or something basis and talk about research articles?
Please.
Yeah, absolutely. It's actually something I take so seriously. And actually here, it's really nice. I work with a lot of practitioners who ask me about research and what does this mean? What are these statistics? And I feel like, well, statistics aren't my expertise, but I can help to some extent.
Right.
But, what does this actually mean for me? And it's kind of I don't want to call it a pet peeve, but it is one of my kind of concerns with the field when these genuinely amazing studies come out, either get misinterpreted or misextrapolated, run with. And, these people who really want information are just not getting the right info. And so I do think all of us academics would love the opportunity. And so I would love the opportunity to move forward with that.
Yay.
To be a part of bringing research to more people. I think it's so important.
So one of the things that we're going to ask people in the comments of this episode, or send me and Stacy a note or something. Mm-hmm. Don't bother Dr. Essler. What studies would you like us to start with? We already have 1 or 2 in the hopper that we're thinking about, but what studies do you want us to go over? Because I know how to read a research study, how to look at what are they actually studying, what do we think they're studying, and oh my gosh, why is their sample size only 5 dogs? Right?
Yeah, that's the biggest critique, right? And then you get to the academic side and they're just like, that's all I could get.
Right. They're like, actually, that was a really big sample size.
Yeah.
And we're like,
yeah. For some students it is. Yeah. Well, you have that kind of idea in the animal world to some extent, where if one animal from that species can do it, like in theory, they all can. So it's like case study style, obviously not necessarily applicable to like detection work, working dog work, but it does kind of come from this idea that if you can show it can happen, necessarily need to have 500 wolves do something.
Okay.
How many captive wolves are there to study? Not 500. So same with primates. I mean, same with working dogs, right? I'm in Cobleskill. If I want to do a study on SAR dogs, how many do you think are within 2 hours of me? And then you have to talk about which ones have different certifications and then continue identifying them. So to give some grace to researchers, it is really hard to study working dogs.
And in doing the paper with Texas Tech, and I know Stacy was involved in a study too, it was literally, we are looking for this subset of a subset.
Yep.
Yeah. And so if you could get 8 to 10 dogs, you were lucky.
Oh, yeah.
And then you have to like make sure that they can do the thing
right.
in the way to be able to collect the data.
Yeah. Yep. And guess what? Oh, yeah, by the way, it's not well-funded research.
Or funded.
At all. Right. So you're asking for citizen science, your canine handlers to show up for free and put up with the restrictions that you're trying to put on the scientist thing. And all of us who are trainers are like, you ask us to do that, and that might screw up my training.
Yep. Yeah. It's really hard. It's really difficult. I think there are certainly studies that could be better, but I don't know a single researcher who wouldn't make it better if they could. We are all working under those constraints. It's also something I teach my students. Same thing when we talk about handlers. When I talk to my students, not completely related, but we talk about like, if you go and work and you're a handler, how many hours a week do you think you can train your dog? And they're like, I'm going to train my dog 30 hours a week. And you're like, okay, with your family and your job. And they're like, oh yeah. I'm like, yeah, you have to have some grace for these people, right? These are real life people. This is not college where you could work 30 hours a week on your dog. And so in the same respect, we're just doing our best. And I do think the intentions are good. And we could also talk about how the peer review process, papers do not start the same way they end all the time.
No, they don't.
So, there's a lot to it, but I do think the intentions are overwhelmingly good. But yeah, I mean, from the handler standpoint, it's definitely, hey, could you come here for free and just let us give you these guidelines that you would never, ever do with your dog and kind of probably don't want to do for me for this research also.
Perfect. Yay. Okay. Dr. Essler, thank you so much for spending all of this time with us.
Yeah.
Amazing.
I know. And
so amazing.
I know. And so to all of our listeners, we're not going to do takeaways because my brain's about to explode.
I'm sitting here. I'm really happy about that because for the last 5 minutes, I've been thinking, what are my takeaways?
Other than, we are training dogs to do all sorts of really cool stuff.
Yeah.
I'm like, I'm just like having to think in a whole different way. That's my takeaway. Is like from a trainer to science, totally different.
Right.
Sometimes.
And goby fish, right?
Yeah.
Goby fish, not being eaten by a dog. So everybody, thank you for listening and go train.
Canine Detection Collaborative.